Hi, everyone. I'm very excited to introduce Jay. He comes to us from the University of Denver, where he did his PhD with Robin Tingatella. If you go to the annual behavior seminars, she gave a talk 2 years ago, which is funny, but it's to remember. Jay even won the W.D. Hamilton Award for Best Dissertation Talk at the 2023 Evolution meeting, which some of you were at, which is pretty cool. So we're going to hear a little bit about that today. He's especially interested in understanding how behavioral interactions shape diversification processes within and between species. So I'm very excited to introduce Jay. Pass it over to Jay.
Awesome. Thank you all so much for having me. I'm so excited to start off our Pop Bio seminar series. And as John said, I come from Robin Tingatella's lab at the University of Denver, and I'm a new postdoc in Kate's lab. But today I'm going to talk to you about some of my PhD work that I did, which I think is a pretty fascinating little evolutionary story all about this guy right here. So, of course, as a biologist, I'm going to start off with a little Darwin reference. So, ever since Darwin originally proposed that we all must be descended from a single common ancestor, biologists have been trying to understand how diversification occurs. So even within one group of organisms, such as these cichlid fishes, there's so much amazing diversity of phenotypes. As an evolutionary biologist, when I look at a phylogenetic tree that shows the relationships between living species, one of the things I'm most interested in is really understanding those very earliest stages of diversification. So how do we start going from one lineage to multiple lineage? And what facilitates that process? Well, one important culprit in initiating divergence can be found in the wonderful world of sexual signals, many that I have shown here. These are very noisy, colorful, and sometimes very strange traits that you don't even have to be a biologist to really fall in love with them. That's why they're in all the nature documentaries everywhere. Often when we go into nature and look at closely related species, the most obvious traits we see that differ between them are their sexual signals, suggesting that changes between these signals may eventually lead to speciation. A now classic example of this can be seen in closely related Laupala cricket species, which have nearly identical morphology, ecology, and life history traits, and primarily differ in a single mating song characteristic, which is the temporal pattern of their songs. And this alone keeps them reproductively isolated from one another. So if the evolution of new sexual signals is important to generating biodiversity, then this naturally leads us to ask, where does this novelty come from? Well, sexual signals are often tightly linked with underlying morphology that are used to generate properties of the signal. Therefore, novelty in signals could arise through the development of new structures, such as this orange pigment that formed a little stripe in this hypothetical fish I drew here, through the loss of structures, or through modification of already existing structures. And these morphological changes could come from various sources, including new mutations, such as in swordtail fish, where some species have lost their really conspicuous tails, you can see here, through hybridizing with other species, as in Amazon mannequins that make hybrids that have brand new color patterns, or through plasticity, like in the scarab beetles, where the horn size is actually nutrition dependent. Unfortunately, studying the origins of new sexual signals and their subsequent diversification is extremely difficult because we're often looking back on the process. So we can take comparative approaches using living species, but we understandably miss those evolutionary dead ends that are peppered throughout the history of populations. Sexual signals in particular often contain various behavioral components that are impossible to measure retrospectively, both in the signaler's behavior, but also in the receiver's response to that signal. For instance, this female bird over here doesn't seem very impressed with the male's display, but we would never be able to know that by looking at, say, a fossil. So during my PhD, I was fortunate enough to have the opportunity to study these elusive early stages of diversification with a recent discovery of multiple new sexual signals. All right, so the main player in our story is this special charismatic little critter here, the Pacific field cricket, which is originally from Australia, but island hopped throughout the Pacific with the Polynesian settlers and eventually landed in Hawaii. Now, let me paint you a little picture here. So imagine you're in this beautiful field in Hawaii, you have the wind in your hair, the sun is beginning to set. You might hear a particular sound that I was going to play for you, but I'm not sure if it's going to work. Let's see. Oh, okay. You can hear that, right? All right, so this sound is a male cricket singing their sexual signal in the form of a loud tonal song. So these songs are generated by specialized structures on male wings that resonate when rubbed together. So structures highlighted here, such as the mirror, harp, file, and scraper, are important in determining the acoustic characteristics of their song. So for example, if you change the size or shape of some of these structures, the resulting song characteristics would then change as well. So males sing these songs to attract females from afar, and then also to entice them to mate once found. And most males have really similar wings and therefore produce very similar songs. And females have preference for those particular signal characteristics. Unfortunately for the crickets, though, there also exists in Hawaii this deadly parasitoid fly that uses those same songs to find its prey. And this kills the singing male, creating this really interesting conflict between natural and sexual selection that is unique to Hawaiian populations of these crickets. Now, around 20 years ago or so, some males actually lost many of the sound-producing structures on their wings, rendering them obligately silent, but protecting them from parasitism. So while being silent actually protects the males from the fly, it also makes it really difficult to attract mates. In 2017, however, my PhD lab discovered a new population of crickets singing a completely different song that sounded in some ways kind of like a cat purring. So we actually named them purring crickets. It's kind of cute. They're actually crickets. They don't look like that, but you know. So this purring song is a lot quieter and had vastly different sound characteristics than that other ancestral song that I played for you. So soon after this discovery, we ran a series of studies where we found that purring song can be used to attract mates, and the parasitoid flies are much less attracted to it, suggesting that purring might serve as sort of like a private mode of communication among crickets. Okay, so everything's just simple, right? Oh, how cool, what a fun evolutionary story. More recently, things have gotten pretty chaotic, even more complicated. So after we went back and worked closely with crickets from sites across Hawaii, we started noticing all of this really unusual variation in both wing morphology and songs that didn't seem to exist before. So some of these songs sounded really similar to the purring song, but others sounded completely different. And these sites have been regularly studied by multiple labs since the 90s. So we were fairly certain that this variation that we were uncovering was indeed new. So what is all of this unexplained variation? So today I'm first going to be discussing my investigation into the origins of the signal novelty across Hawaii, where I characterize the new variation and determine the morphological mechanisms that are underlying the song differences that we see. Then I'm going to focus on just one of these novel signals and ask a series of questions, including where it's spreading to, and also whether the signal is starting to diverge across populations. And lastly, I'm going to be diving deeply into a single population, measuring evolutionary change in real time, and learning how selection is shaping that evolutionary change. Okay, so to start, I conducted that initial investigation into the signal novelty. So we hope that by looking at both songs and wings of these sound-producing males, we could identify whether or not distinct phenotypes or morphs, as I'm going to refer to them today, exist, learn what mechanisms are responsible for song differences, and determine how receivers are responding to the new stimuli, which will inform us how both natural and sexual selection might be impacting these phenotypes. So doing this now and tracking corresponding phenotypic and fitness changes over time might give us insights into how new sexual signals arise, spread, and diversify. Okay, so to do this, we traveled to Hawaii and recorded songs and photographs wings from 6 different sites across the islands. And back in the lab, conducted some in-depth song analysis to measure 10 different song characteristics, capturing amplitude, which is essentially how loud the sound is, temporal pattern, which refers to the sound pattern over a period of time, and frequency, which is essentially the pitch of the sound. So for future reference, this here is called a spectrogram, and this shows the distribution of sound frequencies over a period of time. And I'm going to show you a couple more spectrograms later. So these acoustic properties that I measured make up many of the important aspects of cricket songs. And I don't have time today to go into all the detailed bioacoustics methods that I use, but this is a big part of all this work. And if anyone's really interested in that, I would love to talk to you afterwards. I got lots to say about it. Okay, so after we analyzed the songs, we used photos from those same set of recorded males to quantify variation in wing morphology. So to do this, we used standard morphometric analysis via landmarking, as well as identified the presence and absence of certain wing structures that are known to be really important in sound protection. So then using both the song and wing data together, which included 36 traits per male, we performed hierarchical clustering to group individuals based on their phenotypic similarity. We felt it was really important to use both song and wing characteristics when making these groupings, since looking at just one type of data might not give us the full story, as we're going to see a little bit later on. So our clustering revealed 3 distinct morphs: ancestral, purring, and a new type of male that we named rattling, just based on what its song kind of sounds like. So when we looked at the songs, we found that all 3 morphs differ greatly from one another. So here's what they sound like. I hope you can hear some of the quiet ones on my laptop speakers, so like really listen. So here's ancestral. Here's rattling. And lastly, purring song. So they're pretty different from one another. All right, when we look at the wing morphology of these males, we see that purring wings differ from and are much more variable than the other 2 morphs. However, surprisingly, rattling and ancestral males seem to overlap a lot in their wing morphology, yet they have completely different song characteristics. So keep that in here. Okay, so we characterized this variation, and now we investigated the morphological mechanisms that are underlying these song differences. And when we look at the actual wings of the 3 morphs, we can see that certain structures, such as the harp and the mirror, are fully formed in ancestral and rattling wings, but reduced or entirely absent in all purring wings. Now, because a cricket's wings are the instrument it uses to sing, it makes sense when looking at these that purring wings would produce a completely different song, right? However, when looking at these, you would also expect ancestral and rattling songs to be the same, but they're not. So what is going on here? Now, on a male ancestral wing, there exists this structure called the file that I have highlighted here in red. And this contains a row of many tightly packed uniform teeth. When these teeth on the file are rubbed against by the male's other wing, this sends vibrations through the wing, and that's what resonates the structures to create sound. So when we looked at the purring cricket file, we found that it contains these same tightly packed teeth. However, in rattling crickets, we found these distinct gaps in the file where no teeth developed. Of all of the wings that we looked at under the microscope, 100% of the rattling wings, but none of the other 2 morphs wings had these gaps. And additionally, these gaps persisted over many generations in common garden and were apparent immediately upon males reaching adulthood, so indicating that these gaps are not environmentally induced. So since this is the only difference that we see in the wings between ancestral and rattling males, we assume that this unique modification to the file is responsible for their different songs that they produce. All right, zooming back out to the full wings here, we next wanted to investigate the mechanisms responsible for purring signal. So earlier, if you remember, I talked about those silent males that had lost all the sound producing structures on their wings, right? So when we compared purring wings to these old silent wings, we noticed that they lost many of the same structures. So we broadly refer to these two wing types as flat wings because they don't have those characteristic big structures that give male wings a lot of their bumpy shape and gloss. So what might differ between these two wings, the purring and silent wings, that allows purring to make a detectable signal and silent males to not? Well, we noticed that this one particular structure over here, called the scraper, appeared to be present on purring but not silent wings. So naturally, I asked, is the scraper responsible for restoring signal in purring males? Now, here's a figure showing which frequencies cricket hearing is most sensitive to. So in gray, you can see how loud a frequency needs to be to elicit a response. So the lower the threshold, the more sensitive the hearing at that particular frequency. It's a little bit counterintuitive than you might think. So us humans, our thresholds are somewhat similar to crickets. So I'm going to play two different sounds, and you're going to tell me which one sounds louder to you. Which one sounded louder, one or two? One? Okay. So the first one didn't sound louder to you. Congratulations, you have very special hearing. We might have a novel trait going on in there. But what's interesting is those two sounds are actually played back at the same amplitude. It's just that our hearing thresholds are different. So the first one sounded louder to us. Okay, so what about these songs? So this red line here is the dominant frequency of ancestral song. You can see that crickets are really sensitive to that frequency. Now, flatwing males with a scraper produce higher frequency sounds than this, while flatwing males without a scraper do still produce sounds that our microphones picked up, but they're at even higher frequencies. So that's a 19 decibel difference, which is a lot between scraper and scraperless males. And this indicates that having a scraper makes calling song more detectable to females. And given how quiet purring is already, having a scraper might very well be the difference between being able to make a sound that can attract females and a sound that can't. So we also compared males from a single location in 2015, when all males were silent there, and 2019, when most of the males had become purring. So we found that males with scrapers dramatically increased in prevalence in just 4 years. This is like about 16 cricket generations, really fast. So this suggests that the regain of the scraper over time likely restored signal in this previously silent population. All right, now that we've resolved the morphology responsible for differences, we could address our third aim of this first project, which was to examine the behavioral responses of both female crickets and parasitoid flies to these new songs. To do this, we ran playback experiments, also known as phonotaxis trials, where we played multiple different stimuli to female crickets, and we would measure their response. This is a reminder that this is how female crickets exert selection on male song. So here's a little diagram of how that worked. So we would place a female on one end in a rectangular arena, which also contained a speaker that was facing the cricket. And then we would broadcast the various stimuli to the female, such as purring, rattling, and ancestral songs, as well as like white noise as a negative control. And each song was played individually for a standard certain amount of time. And then we'd record several different measurements to determine the female's attraction to the stimulus. We then ran similar phonotaxis trials using the parasitoid flies. So for this, we use the same set of stimuli, but our methods had to be a little bit different because of how the fly searches for potential hosts. So since it's a fly, it flies. So you can't just put it in a rectangular arena, it will leave. So instead we did these all in mesh butterfly cages, and we had the fly at the top and the speaker at the bottom, as opposed to like this. So kind of just flip it. All right, so what did we find? Well, unsurprisingly, ancestral song was still very attractive to females, with nearly 80% of them exhibiting positive phonotaxis when it was played. This makes sense in long time evolution honing towards these songs, right? But what about the novel songs? So purring did end up being mildly attractive to females. So just under about 20% of them were attracted to the purr, which might not sound very high, but it is better than nothing. And purring was statistically more attractive than white noise. So this indicates that purring males should have more mating opportunities than silent males. All right, now what do females think of this new rattling song? Well, it turns out that rattling is pretty effective at attracting females. It's still not as effective as the ancestral song, but clearly this signal can be used by males to attract potential mates. Now, what about pressure from natural selection? What do we think the fly thought about these new songs? Well, it turns out that they are not attracted at all to these other songs. So even at the really close distances that these trials were run at, none of the flies responded positively to either purring or rattling song, while all of them were positively attracted to the ancestral song. All right, so what does this mean? Well, this suggests that purring and rattling serve as private modes of communication among crickets, both of them. And since their songs are created using completely different morphology, here we have an example of multiple solutions to the same problem of attracting mates while also avoiding fatal parasitism. Calling back to this image I showed you earlier with the hypothetical fish, we now have an example in this single system of signal novelty via the gain, loss, and modification of morphological traits. All right, at this point, we're going to move on to the second portion of the talk. We're going to look at geographic variation and spread of one of these novel morphs. So before we get into this, we need to know where these morphs are located. So as you can see, there is a large amount of morph diversity across Hawaii, with different morphs present at different locations and in various proportions. Two things that might be kind of noticeable to you all. So first, what's going on with this Hilo population over here? It's the only place that doesn't have purring crickets, that has this other thing going on. Just put that on the back burner. We'll talk about that in a minute. So second, and probably the most noticeable thing you may see, is that purring is now really widespread, present at all but one of our 6 study sites. So at 4 of these locations, purring is now the most common morph, and it even appears to have gone to fixation at this Manoa site. And we know that at Manoa, there were at least 50% ancestral males as recently as 10 years ago. So this is remarkably fast evolutionary change that we're observing in these populations. So if purring is already so common at 4 different places, is it the same at all of these places? So remember, these are really small island populations that are relatively isolated from each other. And given the extremely rapid evolution we've already seen in the system, it doesn't seem too far-fetched that purring may have already started to diverge. So do purring wings and songs differ across these populations? Well, we found that wings do indeed differ. So these principal component axes represent the complex variation in wing characteristics that we measured. So you can generally tell that these wings differ because the ellipses don't fully overlap and also have different shapes. And these wing differences included some of those important structures that I talked about earlier, the harp and the scraper. So you can see on the left, harps differed across populations. On the right, we had scraper proportions differed across populations as well. All right, so wings differed. What about songs? Well, when we compared the song characteristics, we found that purring males had differed in both long distance and short distance courtship songs. So again, you can see that the shape and orientation of the ellipses differ by population, and they don't all fully overlap. Interestingly, courtship songs appear to differ more so than calling songs, despite being generated using the same wings. So we don't yet fully understand what is going on here, but we are investigating it in an emergent side project. So stay tuned in 2 or 3 years, probably. All right, so for those who are interested in visualizing the songs using the spectrograms, you can see in these spectrograms, the loudest frequencies, like the darkest ones, and their distribution vary quite a bit among purring populations. So we found that across Hawaii, purring males already differ in their sexual signals, despite the morph arising really recently. So will purring continue to diverge? And could this eventually lead to speciation? Well, we know from other species, like the Laupala crickets that I mentioned in the beginning, that even small changes in a sexual signal between population can lead to speciation. So perhaps this could also happen with purring males. This is almost always facilitated, though, by coupling of these divergent signal preferences or signal changes with receiver preferences for local aspects of that signal. And our previous investigation of female preferences for purring song variants showed that at the moment, females do not prefer certain purrs over others. So this type of reproductive barrier arising in the near future seems pretty unlikely. It will be interesting, though, to continue to monitor signals and receiver preferences in these populations to see if eventually females do start developing preferences for local song characteristics. All right, back to our map here. So if you remember a few minutes ago, I said we would come back to this helo population where there seemed to be more diversity in morphs in this other category. So this is where we're going to move into the final part of the story I'm sharing with you today, where we dive into a single population to try and deeply interrogate the evolutionary dynamics that are going on there. So at this point, I've shown you that there are at least 3 new morphs, one that is completely silent and 2 that we discovered that produce novel signals, each using completely different morphology. But wait, there's more. Since our discoveries, 2 new morphs have been found by another lab called small wing and curly wing. And these males also have altered wing morphology, and as you expect, therefore produce their own altered sounds. One important thing to note, though, is that the genetic underpinnings of most of these morphs are not yet fully understood. As I mentioned earlier, for rattling, though, we do have several lines of evidence across multiple labs suggesting that morph differences are not determined by the environment and that they are strongly heritable. So in this helo population, we found that several of these morphs all live in the same fields and therefore compete for mate acquisition and survival. This gives us a remarkable opportunity to study the evolutionary dynamics in a single location in real time. So remember, this is the exact type of scenario that may have happened many, many times throughout the history of populations that we miss only when we're looking back on the process. So will some of these morphs end up being evolutionary dead ends like we see over here? Why might this occur? Why might it not occur? So in order to answer these questions, we first track changes in morph proportions in the population over time. One morph might outcompete the others, or polymorphism might end up being maintained long-term in the population. We next measured the relative performance of each morph in the context of the primary selection pressures that are facing male crickets. These pressures being sexual selection from females and natural selection from the parasitoid flies. We then attempted to confirm these performance results by rearing males in the lab where there's no natural selection pressure from the fly, and then comparing morph proportions between lab and field. All of this will hopefully allow us to map morph performance onto changes in morph proportions that we found in the wild and infer which of these two pressures is shaping evolutionary change in the population. Because there's been a lot of evidence that the fly drove the initial evolution of silent crickets 20 years ago, we hypothesized that natural selection pressure from the fly will likely be driving the evolution that we see in this population. There's also been a growing body of literature suggesting that predation can be a really important facilitator of diversification. And these studies have been conducted using manipulative experiments or modeling approaches, and we try to add to this to see if it holds true in a natural population. All right, so we systematically sampled males from the population over a 3-year period, which is approximately 12 cricket generations. And what do we find? Well, between the first 2 time points, we found an increase in rattling males, you can see in blue, and a slight decrease in the ancestral and small-wing males. But by the 3rd time point, we noticed the arrival of the curly-wing morph that wasn't even in the population before. So you're probably all familiar with a major global event that prevented us from sampling for a full year. But when we returned, we found that the newly arrived curly-wing males had increased in prevalence while rattling males decreased, and then ending with a slight deviation in the trends that we had observed over the 3-year period. So overall, we found that morph proportions changed over the course of the study, suggesting rapid evolution of sexual signals in the population. Curlywing arose or arrived, we're not sure, sometime after summer 2019, you can see here, and then rapidly increased in prevalence, coinciding with a general decrease in the other morphs. So clearly, morph proportions are not at equilibrium in this population. Okay, so now that we tracked changes in morph proportions over time, we measured the relative performance of each morph in both mate attraction and fly protection. So I'm going to have a table here that I'll be filling in over the next few minutes for you all. So females and flies assess male crickets as suitable mates and hosts in both long distance and short distance contexts, as I discussed a little bit earlier. So we conducted 4 different experiments to resolve the relative performance of each morph in all of these scenarios. So how does each morph balance the conflicting sexual and natural selection pressures? Well, we first confirmed our prior work, which showed that ancestral males were still by far the most attractive to females in a long-distance context. So in 4-way competitive phonotaxis trials, females chose ancestral song 70% of the time. So ancestral is clearly still preferred by females over novel songs. So to compare female responses to just the novel songs, we wanted to understand how the novel morphs were being assessed in a competitive context using a round-robin format, where each female was given all combinations of 4 stimuli: rattling, small wing, curly wing, and silence for a negative control. All right, so what do we find? Well, we found that novel songs differed in their relative attractiveness to females in a long-distance context. So you can see on the y-axis here, this relative attractiveness score. This is a very complex metric, but all you need to know is that a positive value indicates disproportionately more attractive songs. Negative value indicates relatively unattractive songs. So as you can see, small wing and rattling songs were more attractive than curly wing songs. And interestingly, curly wing attractiveness did not differ from silence, suggesting 1, that curly wing song was terrible at attracting mates, and 2, that it might not actually serve as a functioning sexual signal right now. So filling out our table here, we can say that morphs differ in long-distance mate attraction, with ancestral males being the most attractive and curly-wing males being the least attractive. Now, what about short-distance mate attraction? Well, we found that in these close-range courtship encounters, morphs surprisingly did not differ in their attractiveness to females. And this was true when looking at both mounting proportions on the left and also latency to mount on the right. Now, at first glance, it might kind of look like maybe some morphs are a little bit more favored over others during courtship, but we didn't find that they actually differ statistically, suggesting if there is sexual selection acting on courtship differentially among morphs, it's likely pretty weak. Okay, so we found that morphs are equally able to attract mates in a short distance context. Now, how do they fare against the fly? Well, using traps in the field that are broadcasting the various songs, we found, not that surprisingly, that ancestral song is more attractive than the novel songs. This is similar to what we found in previous work when we compared ancestral and purring songs. So novel songs did not differ from each other, regardless of whether there was a competing ancestral song being broadcast. That's the difference between these two here. Now, despite this, it is interesting to note that of the novel morphs, we did catch at least some flies to rattling and smallwing, but never to curlywing song. So to summarize, ancestral males are least protected from parasitism at long distances. So what about fly preferences in a short distance context? Well, using those same close range butterfly net phonotaxis trials that I described earlier, we found that that morphs actually did differ in their attractiveness to flies. And this time, even the novel morphs differed in our measures of positive phonotaxis. So on the left side, you can see distance traveled towards the speaker. On the right side, proportion of speakers contacted. The ancestral was the most attractive, followed by smallwing, rattling, and lastly, our good friend curlywing, which did not differ from silence. So in summary, morphs differed in their protection from parasitism at close range. So now that we measured morph performance, we could compare morph proportions between lab and field with and without predation from the fly. So when we collected females in the field where we had already determined morph proportions, which you can see here, we had them lay eggs, and then we brought back those eggs and had them reared in the lab under standardized conditions in common garden. As soon as they reached adulthood, we phenotyped all the males and determined morph proportions in the lab. We also looked at morph proportions in the field from that second generation so that we could see whether proportions changed depending on whether the fly was present. So we found that morph proportions did not significantly change between generations of males in the field. However, proportions did change when males were reared in the lab without parasitism. One explanation for this is that in the wild, ancestral males might be acquiring more mates, but are surviving less as adults, while curlywing males are being mated with less, but are disproportionately surviving. If this is true, it would confirm the previous performance results, where we found that ancestral males are most attractive to both female crickets and flies, and curlywing males are least attractive to females and flies. However, I do want to approach this conclusion with some caution because there are other ways that we could get differences between lab and field. Because as I mentioned, the genetic architecture underlying these traits isn't fully resolved. And other mechanisms like plasticity could be contributing to the difference that we saw between lab and field. All right, lastly, we aim to infer which of these two primary selection pressures may be driving the evolutionary dynamics we observed over the course of the study. To answer this, I mapped the relative morph performances that we found, which I summarized in this table, onto the changes that I found in the population over time. So if we look at sexual selection, ancestral males are the most attractive to mates, while curlywing males are the least attractive. So if sexual selection were driving patterns of morph proportions in the wild, we would expect to see ancestral males in red increasing over time and curlywing males in yellow decreasing. Instead, we see the opposite. So when we look at natural selection, the morph that is most protected from parasitism at the time seemed to increase in prevalence, beginning with rattling, followed by curlywing after its arrival. So all in all, this suggests that of the two major selection pressures that male crickets face in the wild, natural selection from parasitism is more likely to have driven the rapid evolutionary change that we witnessed in the population. All right, so given this, will curlywing eventually outcompete the other morphs? Well, it's possible, but morph proportions do appear to be quite dynamic, and the fly's importance in shaping the population could end up changing in the future, and polymorphism could end up being maintained long-term. So this work here capitalized on an opportunity to learn how evolution's playbook unfolds during the early stages of diversification and to follow along as it's being written. I think it'll be really interesting to conduct similar real-time studies in other locations across Hawaii that have different combinations of morphs to learn whether selection shapes populations in a repeatable way. For instance, in Manoa, which is a population on Oahu, this place has all purring males. We know there is at least a little bit of gene flow among populations. So what would happen if rattling males, which are more attracted to females than purring males, but also are pretty protected from the fly, what if they migrated to Oahu? How then would this playbook unfold? So I really believe that the tractability of the system makes it a really powerful microcosm for answering these and other such fundamental questions in biology, and why this work excited me so much over the course of my PhD. Okay, super briefly, I know everybody, it's like almost 5 and people want to go, but I just want to end here talking a little bit about what I'm planning on doing here at Davis as part of Kate Laskowski's lab. So I'm sure, as many of you know, Kate's lab focuses on studying animal behavior using the clonal Amazon Molly system, and in particular, answering questions about individuality. So I'm really interested in using this fascinating system to learn more about when variation in personality develops during life, and investigating how this variation may contribute to the early stages of diversification. And I only recently started on this work, so if anyone has any thoughts about this, like I would love to hear them. I'm very excited about it and have a lot to learn. All right, I have so many people to thank. All my co-authors, especially my awesome advisor, Dr. Robin Tingatella at the University of Denver, 3 rock star co-author undergrads, Aziz, Brianna, and Hannah. They're amazing, integral, helping me collect data and helping with some analysis, as well as lots of people who ended up collecting data, some part of the project, especially field samples when we were in Hawaii. Huge thanks to everybody. And of course, this work would not have been possible at all without any of the funding and research support from all these sources. So yeah, and with that, thank you all so much for listening. Really appreciate you giving me this chance to tell you about my work. Thanks. All right. Yeah.
Yeah, I was wondering if you had any idea of the fly population numbers, because
Mm.
on your proportion plot, you see the curly wing one go up.
Yeah.
And then after that, you then see the ancestral morph go up right after that. Do you have any idea of whether or not the fly population plummeted?
Yeah.
Because of that curly wing morph not being parasitized?
Oh, that is a fantastic question. We do have those data somewhere, and like that is something we totally want to dive into, because that could completely be affecting these dynamics, right? Like all of a sudden you have a crash in the attractive morph, and then the crash in the flies, and how eco-evo dynamics work, right? There could be like a lag, right? So yeah, we do have those data, and I would be super fascinated to look into that. Yeah. Yeah.
So I think in the middle of the presentation, we showed the proportion of the different morphs across the islands. I couldn't help but notice the biggest island have the highest diversity of
But the Big Island, yeah.
Yes, so I'm wondering, like, what was the migrational history of these? Because they start in the Big Island, I'm wondering, like, what they like.
Yeah, that's a great question. So, well, a couple of things to note. These are all from particular populations. So the island size, we're not like measuring all across an island. So it's not like the Big Island has the most crickets or something. Although in this case, it kind of does, just that's by chance. Yeah, so it's actually, that hasn't fully been resolved, but there's been some recent work out of Nathan Bailey's lab that's shown kind of the relationships between the islands on a little bit of a cursory level. The Big Island was not the first one colonized. So it's a little bit like Oahu, Kauai, Big Island. It kind of jumps around in a way that you might not expect. But that just could be because of the gene flow, how things work. Like, a boat goes to one place and it happens to be in a field or whatever. Yeah, that's a good question.
That same plot, do you have an explanation for the population that still had a large proportion in ancestral north?
Yeah, so very interesting. So it's possible that the flies hadn't arrived there until later. It's also possible that something about that population that might favor having ancestral males. Something I've really been interested in is thinking about like the physical kind of structure of the field that they're living in. So if you had a field with really narrow corridors where you wouldn't have to kind of travel that much to randomly bump into a female, that might even more favor these like attenuated morphs that aren't attractive. Whereas like places with like big open fields, you might expect be harder to find mates that way. And so you would have like that go a little bit in a different way. But yeah, it's honestly possible because of the timescales that we're talking about. It could just be chance. Like maybe Perrine didn't show up there until like right when we started the study, but hasn't had time because it does take years, right? But yeah, that's a really good question. We're not entirely sure. Yeah.
I know, like, in what I don't know if these crickets are they more in the native forest or are they kind of in the disturbed?
Yeah, these are not like native. They live in like college campus fields. Like there's Oh yeah. So
yeah, that's my question is
yes.
Solution. There's a bunch of super loud.
Yeah.
Noise,
Oh yeah. Yes.
So have you seen kind of noise pollution have an effect on song?
Yeah, so this is really interesting. There are a couple other people working in my PhD lab on that. They're a little bit more interested in the anthropogenic stuff. I will say that, so the kogi frogs, in terms of the islands that we study, they're actually only on the Big Island. So they're only a problem there, but like there's all this morph diversity there. So it's kind of like counterintuitive. But earlier studies actually have found that females have no attraction to the kogi frog and that it doesn't interfere at all with their ability to find mates. So because of the way the frequencies are, the kogi frog frequency is around like 2 kilohertz, and that's pretty low compared to most of cricket song. So it doesn't end up interfering. What it does interfere with is me when I'm trying to analyze the songs, and I'm like here we go again. Like it's just, yeah. So I have to like work around that. That is very difficult. But that's a great question. In terms of other anthropogenic stuff, these really are like, living in places that are very urbanized, and there's some locations that are more so than others. But we don't quite understand why that's fine for them. They seem to not care at all. And in their homeland of Australia, also, live in like near highways, and so they're just like they're crickets. They do whatever they want. Yeah.
Yeah, I know you said that the genetic base is not fully characterized. I'm very curious if it's like markers.
Well, okay, so yeah. So yes.
Yeah, because these different mating calls
I should have this up.
What seemed like really discrete aspects of the wing.
Yeah.
That to me suggests that it could be like one or just several loci.
Yes. So okay, so the original silent mutations have been resolved. So that's why I said most. The other ones, they weren't discovered until the earliest was 2017. So there's just like hasn't been enough time to figure out like where they are, right? But the silent one thing that's really interesting about the silent morph is that recent work has shown that it independently evolved 3 different types. So on 3 different islands, the silent crickets just are their own thing, even though they look really similar. Definitely an excellent situation. I don't think anything else you know about these. something going on with double sex. I think people are very highly suspicious of that. But in terms of the rattling and like purring and these other ones, that work is being done right now, but we really, like, I don't want to guess because it could be something that's right. And one thing that I didn't get to say, which actually is really cool, I think regarding that is, especially in philo, you have all these different morphs. Very recently, like pretty much after this study was conducted, we started noticing some males that are like compomorphs, hybrids. Like they have their wings have characteristics of like curly and smaller. And we're seeing those a lot more in the lab now. So something's definitely going on in terms of they can be independently segregated. Yeah.
Wondering about the female pregnancy, that's changed at all over time.
Yeah, so we've only been looking at well, I shouldn't say only. The longest morph we've been playing back to them is the purring song, because that's the one that we discovered first. And so far, we don't have any evidence that that actually has changed in terms of liking certain purrs more than others. Now, this data set is large and it's ongoing. So by the end of like the long-term study, it could end up that maybe they're just more responsive to purrs overall, as opposed to liking particular purrs, which is really interesting because that is still a change in preference, right? In terms of rattling, who knows? It's interesting because that right now is only in one place. So maybe that will become kind of a local thing, right? Maybe they start liking aspects of rattling song more than in other locations. Yeah.
I'm just curious, and I'm kind of confusing myself, so
Oh,
I'm
sure.
just wondering what you're thinking. So in your talk with these crickets that reproduce sexually, you think of this interplay between sexual selection and natural selection.
Yeah.
But now you're working in a system that's clonal.
Yeah.
And you see this variation. So
Yeah.
given that these organisms are clonal, what are the two mechanisms that might be interplaying here? Like, is it still predation? Like, you had this whole slide on like predation driving variation. And then like, are they social organisms as well? Like, do they live in schools? And so they'd be like social versus predation, or like, how do you look at this system that you work with now, which is now clonal?
Yeah, I mean, that's a great question. I think it makes it a little bit different when asking questions about sexual selection. The system, though, the species is clonal, but the system isn't fully clonal. So our Amazon mollies they're actually sperm parasites. So they're reliant on these other fish that live near them that are closely related. They're gonna parasitize the male sperm. So there's definitely like a mate choice system going on because, you know, they're essentially trying to like trick these other species males into providing sperm for them, but then the males aren't necessarily getting a benefit out of that, but then they might be because it makes them more attractive to the other females. So there is definitely like a, you know, sexual mate choice thing going on there. It's just really different. Yeah, and that's a really interesting question. I mean, I think that's something that I'm trying to figure out because I'm really interested in those dynamics, and some of those just work really differently in the models. Yeah. Thanks.
Maybe last question.
Oh, yeah.
Please.
Sure, Max, you can ask me later.
Thanks for the talk. That was super great. I'm thinking a little bit about, you know, Fisherian dynamics and coevolution between preference and sexual displacement, right? And so I'm thinking, you know, about whether you guys have established any populations that are strictly, you know, one display type, and then seeing if the females that result from that population kind of develop preference with time.
You're talking about with crickets.
Yes.
Yeah, okay. Yeah. I was like, well, okay, so that's something I would absolutely love to do, like a long-term project, like setting up like mesocosms and see if we can observe rapid evolution over time. I think we could, given the rates that are going on, especially if we're setting them up in a situation that's really favoring them, you know, very controlled. Yeah, I think that's a great idea. I bet we could find it, and I think people should do it.
Well, thank you everyone for coming and listening to the